In this video, we're going to talk a little bit about the physiology of male reproduction, including some particular concerns, some particular considerations specific to forming sperm cells as opposed to forming the female gametes. We'll start with sperm cell formation, and in order to understand what we're talking about with sperm cell formation, we need to talk a little bit about the structure of individual sperm cells. The sperm is kind of a weird cell. It is basically just a nucleus with a delivery mechanism. So in the sperm cell you have the head, which contains the nucleus. It also contains a small sac of enzymes called the acrosome. The acrosome is filled with enzymes that are essential for penetrating the female egg and actually allowing fertilization. Behind the head you have the midpiece, and the midpiece contains a set of mitochondria that power the flagellum, and the flagellum is the propulsion mechanism of the sperm, the so-called tail of the sperm. So looking here, actually under scanning electron micrograph, head, midpiece, flagellum. Sperm have very little cytoplasm, and the only part of the sperm cell that actually makes it into the egg at fertilization is the head. I want to show you a little video of sperm cells in action. We're actually looking at mouse sperm, and it's going to look a little bit different than the human sperm you see here because the head of mouse sperm cells has a hook shape. It's shaped more like a comma. Also, you can actually see the movement of the flagellum in this video because the sperm have been placed in a viscous fluid. So it slows down. Since the fluid is so thick, it slows down the movement of the flagellum to a point where you can actually see the sine wave-like motion of the flagellum. There you can see the little hook shape and the sine wave shape of the beating flagellum. The process of forming mature sperm cells is called spermatogenesis, and there's two parts to it. The first part is meiosis, the actual division of the sperm stem cell, the spermatogonium. The second part is spermiogenesis, and spermiogenesis is where you take the haploid cells formed from meiosis and you cause the changes in shape, the changes in form that make for a mature sperm cell, ready to be ejaculated and fertilize an egg. So there's some terminology. As I said, the sperm stem cell is a spermatogonium. This is the initial cell, the initial diploid cell that becomes 4 sperm cells. And it's referred to as a stem cell because when the spermatogonium first divides by mitosis, one daughter cell, so mitosis, it makes 2 daughter cells, one daughter cell remains as a stem cell, remains as a spermatogonium. The second daughter cell goes into meiosis and becomes the spermatids. So it becomes what's called a primary spermatocyte as it enters into meiosis 1. At the end of meiosis 1, it's referred to as secondary spermatocytes, and these cells are actually joined by a little cytoplasmic bridge. The secondary spermatocytes go into meiosis 2 and become what are called early spermatids, all still joined by a cytoplasmic bridge. It's the spermatids that go into spermiogenesis and convert to sperm cells that we're all familiar with. So there's a progression from spermatogonium to primary spermatocyte, secondary spermatocyte, early spermatid, late spermatid, to finally the mature spermatozoa. This is all happening in the seminiferous tubule, and you're seeing a cross-section of the seminiferous tubule here with the lumen and then the wall of the seminiferous tubule around that. Now, meiosis occurs on the outside of the seminiferous tubule, and as the sperm cells mature, they get pushed in towards the center. So the more immature cells, like the spermatogonia, are on the outside of the wall of the seminiferous tubule, and the mature spermatozoa are in the center, in the lumen of the seminiferous tubule. Once you have the early spermatids, the process of spermiogenesis, going from this relatively normal-looking cell to the mature sperm cell, takes about a month, about 24 days. And there are a few things that have to happen in this process. First, you need to have the formation of the acrosome. Remember, the acrosome is that little bag of enzymes that allows a sperm to penetrate the egg. You also need to lose the cytoplasm, so excess cytoplasm is gotten rid of. You need to grow the midpiece and the flagellum. So you're seeing here early spermatid, relatively normal looking, still relatively normal looking, starts to extend the flagellum, the microtubules that form up the flagellum. The acrosome starts to spread around the nucleus, starts to take on sort of a distinctive shape as the flagellum continues to grow. The mitochondria start aggregating around the base of the flagellum, forming the midpiece. And then the excess cytoplasm is eliminated until finally all you have left is the mature spermatozoon. Now, there are two types of cells, apart from the cells that are actually directly involved in making a mature sperm cell, apart from the spermatogonia and the intermediate cells. There are two types of cells found in the testis that are absolutely critical for supporting the process of sperm formation and regulating the process of sperm formation. I mentioned the interstitial cells, also called Leydig cells, in the male reproductive anatomy video. These cells are referred to as interstitial because they're located between the seminiferous tubules. So here's one seminiferous tubule, here's another seminiferous tubule, here's another seminiferous tubule, and here are the interstitial cells. They tend to cluster near blood vessels between the seminiferous tubules, so they're conveniently located for the hormones that they produce to be taken up into the blood. The hormones they produce are testosterone and other androgens, including DHEA. DHEA stands for dehydroepiandrosterone. DHEA is a weak androgen that can be converted to testosterone. The interstitial cells are very active in the fetus, where they promote male genital differentiation. So they're important in the initial development of a individual who is not just chromosomally male, but who actually presents physically as a male. They then go quiescent during childhood and get reactivated in puberty. The second type of supporting cell we find in the testes are the nurse cells. Nurse cells are also called sustentacular cells and Sertoli cells. And these guys are actually located in the wall of the seminiferous tubule. They're these big, long cells. I've outlined a couple for you here. These cells, they basically surround the spermatogonia. And one of their crucial functions is to promote sperm formation. They promote both the mitosis of the spermatogonia, the meiosis of the daughter cells to form spermatocytes, and they promote spermiogenesis. They provide necessary nutrients and stimuli that the sperm-forming cells need in order to develop sperm. And these guys they phagocytose and get rid of the excess cytoplasm that's lost during spermiogenesis. These guys are responsible for the blood-testis barrier. The blood-testis barrier makes the testes an immune-privileged site, so that the reason why it's important for the testes to be an immune-privileged site is because there are all sorts of genes being expressed, all sorts of proteins being made during sperm cell formation that are not made anywhere else in the body and that are not made at any other time. So these are proteins that may potentially be attacked by your immune system. except that you have this blood-testis barrier so that the immune system does not patrol the testes and doesn't see these unusual antigens. The blood-testis barrier also gives some separation between the luminal fluid, the fluid in the lumen of the seminiferous tubules, and the blood plasma. This separation allows the luminal fluid to be different from the interstitial fluid. The luminal fluid is higher in androgens, estrogens, potassium, and amino acids. Nerve cells are also important regulators. They both help promote sperm cell formation, and they produce a negative feedback that regulates and slows down sperm cell formation. They secrete a molecule called androgen binding protein. Androgen binding protein basically holds testosterone in the seminiferous tubules. That's why it's called androgen binding protein. It binds up testosterone and holds it there so that you have a higher concentration of testosterone in the seminiferous tubules. And then that allows that testosterone to promote sperm formation. They also secrete a hormone called inhibin. And inhibin is released in a way that's directly proportionate to the number of sperm formed. So the more sperm you have being made, the more inhibin you have made, and that slows the sperm formation down so that you don't end up with too many sperm being made. There are a number of hormones involved in regulating spermatogenesis, not just testosterone and inhibin that we talked about before. This slide gives you a complete list of the hormones involved and a quick summary of what they do. But to talk a little bit more, to talk through it, I'm going to flip ahead to the next slide, which shows a visual representation of how these hormones interact. So on this slide, you're looking at a visual representation of the hormones. Arrows in red represent a stimulatory relationship, one hormone stimulating another. Arrows shown in blue represent an inhibitory pathway. So you start up here with GnRH, gonadotropin releasing hormone, and that's released from the hypothalamus. GnRH triggers the release of FSH and LH from the anterior lobe of the pituitary. So GnRH triggers FSH and LH. Now, FSH and LH, although they're named after their roles in female reproduction, they also have roles in male reproduction as well, in the formation of sperm cells. FSH, if you follow this pathway down, FSH acts on the nurse cells. Okay, so FSH promotes the function of the nurse cells, triggering them to release androgen binding protein and promote sperm formation. Luteinizing hormone, LH, acts on the interstitial cells, the Leydig cells, tells the interstitial cells to secrete testosterone. Testosterone maintains the male reproductive system. It promotes sperm cell formation. It also is responsible for male growth and development of secondary sexual characteristics during puberty in males. You've got a summary down here of the effects of testosterone: maintaining the libido, stimulating bone and muscle growth, stimulating male secondary sex characteristics like a deepening voice, like development of hair, as well as maintaining the accessory glands and organs of the male reproductive system. Testosterone also has a promotional effect on nurse cells, a stimulatory effect on nurse cells. So testosterone supports the nurse cell functions. Testosterone has a negative feedback loop in it as well. If the testosterone levels get too high, testosterone actually gets into circulation. Well, it's always in circulation, but high levels in circulation shuts off GnRH. And so when GnRH is turned off, the levels of LH reduce and the levels of testosterone come back down. And then once the levels of testosterone drop, this inhibition is removed, and you can maintain a stable level of testosterone that way. We have another negative feedback loop here, and that's the production of inhibin. As I mentioned, you get more inhibin released the more sperm cells you have made. So inhibin is released by the nurse cells, and inhibin acts on follicle-stimulating hormone. So inhibin, when you get high levels of inhibin, it suppresses follicle-stimulating hormone, thereby suppressing nurse cell form stimulation, and then slowing down the rate of sperm formation. Inhibin is also thought to maybe have an effect on gonadotropin-releasing hormone as well, but that's not as certain. As with pretty much everything in homeostasis, it's all about having enough, but not too much. You don't want to overdo it on sperm cell formation. You don't want to overdo it on testosterone levels. So you have these negative feedback loops through inhibin and through testosterone itself that ensure that under normal conditions, sperm cell formation and testosterone production don't get too excessive. Okay, so these are some of the questions that you should be able to answer after studying this video, particularly after studying it together with the male reproductive anatomy video. You should be able to distinguish between spermatogenesis and spermiogenesis. So remember, spermatogenesis is the whole process. Spermiogenesis is just the maturation of the spermatids. You should be able to describe the structure of a sperm cell. You should be able to explain the functions of the different parts of a sperm cell. You should be able to describe the functions of the interstitial cells and the nurse cells, and of these hormones in regulating male gamete formation.